Hello, good morning, Naboja.
Good morning, everyone. Good afternoon, wherever you are.
Hello. Yeah, is it audible?
Yes, I can hear everything. I hope that you can hear me well.
Yeah, you can hear you. Naboja, just 2 minutes. We will wait. Can you wait? Just 2 minutes.
Yes, no worries. Yes.
Just. I'm meeting myself. Eh?
Never. Yes, great. Thank you.
స్టాఫ్ లంచ్ వచ్చి స్టాఫ్ అక్కడ అందరూ అసలు పొద్దున్న తెలుసా నా ఫీల్ ఆయన నన్ను చూస్తుండే నేను ఆయన చూస్తుండే.
Hello, Dr. Nebo.
Hello.
Hello, Dr. Nebo. Is it audible?
Yes, I can hear.
Yeah. Thank you. Once again, Dr. Nebo, to be to accepting our invitation as one of the eminent speaker for our this 2 days international symposium. Now, this 2 days international symposium, it is sponsored by CSIR Government of India, and here we have few speakers from India and few speakers from out of the India. And once again, thanking you for accepting our invitation. So, Dr. Neboza Pavlik is an assistant professor at the Department of Pharmacy, Faculty of Medicine, University of Serbia, where it is pharmaceutical technology. His experience and research depends on the experimental pharmaceutical, sustainable drug design, translational drug research, and his mainly work on the bile acid as the therapeutic drug, transport promotional mechanisms, and he is very experienced in micro and nanostructured drug delivery system. It including the emulsion, lipid and polymer based matrix carriers, and oral drug delivery system. And he has underwent international research stays at Kyoto University, Australia, University of Malaya, Malaysia, where he experienced in microfluidics and targeted drug delivery. And to his credit, he authored more than 60 peer-reviewed scientific articles in SCI journals, having more than 2,000 citations with H index of 22. And he is an excellent reviewer and guest editor for several international journals, especially in the pharmacy and pharmacology. He also the recipient of several prestigious awards. He is also the member of several professional and scientific society, including International Pharmaceutical Federation (FIP) and Marie Curie Alumni Association. He is very fluent in English and has working knowledge with German and Spanish language. And thank you, Dr. Pavlik. And this is up to you now. You can start, please. I think you are muted. You can unmute now.
Can you hear me now? Yes, thank you. Thank you once again for the invitation to be here for the second time after the first successful symposium. So it's really my pleasure to be here now and to present some part of my research, hopefully some next time also in person in your beautiful country. So yes, my presentation and my today's lecture will be focused on rheology and generally on development and characterization of emulsion systems stabilized by natural constituents, mostly protein isolate, specifically soy protein isolate, and also of natural gums. We investigated mostly xanthan gum and guar gum that we will see later can form really interesting drug delivery systems that is in the focus of the research of our research group. So first, I would just say a few words about me and my team. I come from Serbia, from southern Eastern Europe, and from the second largest city of Novi Sad in the north of the country. I will just say that about my city. It's a university city, very lively, and you can see some photos here. And it was really it's a nice city for young people. It was the European capital of culture a few years ago, so European youth center cities, and that's something that you can see here. And as I said, it's a university center. It's second largest university in our country. And here is my institution, where I work. It's Faculty of Medicine, School of Medicine, actually, and up to the right is also the Department of Pharmacy, where I work, where is my lab and my office. So yes, you can see it's the Faculty of Medicine was founded around 70 years ago, and our Department of Pharmacy is 26, 27 years old now. And after these years of our development of our institution, we also managed to develop our laboratories. And as it was said in the introduction, my main focus is pharmaceutical technology, drug formulation, design, and characterization. And now we are mostly focused on drug delivery systems, on their rheological characterization, also characterization in terms of particle size, and also some texture analysis, and everything. We work on different drug delivery systems for peroral, topical administration, also for specific administration routes. So that's basically what I just wanted to say, a few words at the beginning. And before presenting the results, I would like to explain why soy protein isolate was chosen and why it represents a promising material for innovative and sustainable drug delivery, which is a topic central to this symposium. So first, I would like to say that soy protein isolate is a highly purified protein fraction obtained from soybeans, which contain around 40% protein on a dry basis. As we can see in this scheme, after defatting and extraction, the resulting isolate contains approximately 90% of protein, and the main protein fractions are globulins, particularly the 7S and 11S components, together with some smaller amounts of albumins. But we will see those two fractions are the most important for us and for using them in drug delivery systems. Soy protein is widely used already, but it was mainly used in the food industry, but now it's more and more used also in the pharmaceutical and cosmetic industries due to its safety and availability, and also with the rising significance of that green pharmacy context that is very important nowadays. And from the drug delivery perspective, it's particularly attractive because it's biocompatible, biodegradable, amphiphilic, so it can interact both with aqueous and lipid phases. Also, it can exhibit multiple functional properties such as gel formation, emulsification, film formation, which is all very, very interesting nowadays in research. And that's also very relevant for designing drug delivery systems. So, as I said, the majority of soy protein isolate consists of two globular proteins, beta-conglycinin, known as 7S fraction, and glycinin, known as the 11s fraction. Together, they represent more than 80% of total soy proteins, and structurally, as we can see here, beta-conglycinin forms a trimer, while glycinin forms a hexameric assembly, which is also important for emulsification, also interfacial properties when developing emulsion systems, for example. These proteins are stabilized by hydrogen bonds, hydrophobic interactions, and disulfide bridges, resulting in compact globular structures with relatively limited flexibility in their native state. But this structure rigidity is very important because it impacts their interfacial behavior and gelation capacity that we will see a bit later, because it is required that partial unfolding or denaturation is required to activate their functional properties in drug delivery systems. What are some main molecular features relevant for drug delivery systems? At the molecular level, this protein isolate contains both hydrophilic and hydrophobic amino acid residues, so it has amphiphilic character. It also possesses reactive functional groups such as amino, carboxyl, sulfhydryl, hydroxyl groups, which allow intermolecular interactions and chemical modifications, which is also very important in order to improve our drug delivery systems. Also, these hydrophobic patches on the protein surface enable absorption at oil-water interfaces, while environmental changes such as pH value or temperature can change only those conformational transitions and slight denaturation that can expose reactive groups and promote intermolecular cross-linking. We can see in these schemes how that unfolding occurs, and as a result of these molecular features, soy protein isolate can form gels. It can be induced by ions, by cross-linking, or thermally induced gels, which we also investigated. Also stabilize, it can stabilize emulsions, generate nanoparticles, and also be conjugated, covalently conjugated with drugs or other polymers. And this multifunctionality actually underlies its versatility in drug delivery applications. So about, of course, always molecular features are connected to physicochemical properties, and we can see that this protein isolate exhibits isoelectric point around pH 4.5 to 5, where the net protein charge is minimal and aggregation tends to occur at these acidity levels. So its behavior is strongly influenced by not only pH value, but also ionic strength and temperature. We can see it summarized in this table, but I will say only shortly. When partially denaturated under controlled conditions, this protein isolate actually shows increased surface activity, improved emulsifying ability, and stronger gel formation. And these changes occur because unfolding exposes, as I said, hydrophobic and reactive groups that enhance intermolecular interactions. So it's very important when developing this kind of drug delivery systems to carefully control these environmental conditions, because it changes functionality and is actually essential for optimizing drug delivery systems based on soy protein isolate. Main technological functional properties are, as I said, gelation, emulsification, water holding capacity, oil binding capacity, film formation, and foaming. We can see some of these. For example, we can see microstructure of thermally induced gels under different conditions, so how they act and how they look like actually under different conditions. Also, we can see how it can form not only a regular gel structure in terms of some matrix system for encapsulation or something else. It can also form edible films that are more and more popular. And also, as I said, because of its interfacial activity, it can form very stable emulsions. We will see later, often in combination with some other biopolymers, such as natural gums. So because of this broad functional profile, this protein isolate can serve multiple roles in drug delivery formulation, including matrix former, emulsifier, stabilizer, and carrier material. I will just say a few words about gelation mechanism, which is very important for this protein. It generally occurs through a two-step mechanism. First, denaturation, but not complete denaturation, just slight denaturation unfolds the native globular structure, exposing those hydrophobic but also sulfhydryl groups that were previously inside this globular molecule. Second, these exposed groups participate in intermolecular interactions such as hydrophobic association, hydrogen bonding, disulfide bridge forming also. And this leads to aggregation and formation. We can see in the second phase we have aggregation, and finally the formation of three-dimensional network. So the resulting hydrogel matrix can entrap water and dissolved or dispersed drug molecules, which makes this protein as a suitable controlled release carrier and also tissue-like biomaterial in drug delivery application. Not only drug delivery, but also because of its high biocompatibility, it's also for cell delivery, also in some kind of tissue bioengineering, and it's also some attractive research topic when we talk about this protein isolate. As I said, soy protein isolate can form hydrogels through thermal or chemical induction, and they contain a large amount of water and therefore mimic the mechanical properties of biological tissues. And there are different systems that were developed for this protein, not only topical, but also injectable, implantable applications, and also drug release from these SPI hydrogels typically occurs through combination of diffusion, matrix felling, and gradual erosion. So we can achieve controlled release. But as I said, because of different environmental factors that are very important for drug release, we can be very careful when optimizing this formulation. Second, large group of formulations that this protein can form are nanoparticles and microgels, mostly also microspheres, different kinds of matrix systems, and these particulate carriers can be produced by methods such as desolvation, pH-induced aggregation, thermal treatment, also ionic cross cross-linking. Those are the most common methods, and this protein is suitable for these technological procedures. These particles, they can be microparticles, nanoparticles, etc., are particularly interesting for oral delivery of bioactive compounds, protection of sensitive molecules such as peptides or proteins, and encapsulation of nutraceuticals. There are many publications on recent publications on developing nutraceutical formulation, and their biodegradability and food grade origin also support safety, of course, and regulatory acceptance. So we can conclude here that these protein nanoparticles can represent a promising alternative, natural alternative, and sustainable alternative to synthetic polymer carriers. Of course, I already mentioned, and in the experimental part, it will be more focused. This soy protein functions as an effective natural emulsifier because it can adsorb at the oil-water interface, reduce interfacial tension, and form, also very important, not only it's surface active, it forms a very stable protective viscoelastic film around droplets. And that's also something that we focused in our research to not only rheology of the whole system, of the whole emulsion, but also interfacial phenomena such as interfacial tension, but also interfacial rheology, because the rheology of the whole system and the interface is not the same, because some part of the protein remains undissolved, so it forms some kind of Pickering emulsions with solid particles around oil droplets, which also contributes to system stabilization. Generally, these protein stabilized systems combine natural origin with functional performance comparable to synthetic surfactants, but also they have more benefits, especially considering their biocompatibility and sustainability. Also, a very important aspect when we talk about protein-based drug delivery systems are the complexes and general interactions with some polysaccharides. I will focus on natural gums a bit later, but generally this protein can interact with polysaccharides through electrostatic attraction and hydrogen bonding, and these interactions lead to formation of complexes such as mixed gels, multi-layer interfacial structures, and also coacervates. We also won our publications now. It's under review right now, but we also investigated the occurrence of these coacervates that are very interesting from the formulation aspect, because their formations sometimes are something that we want, sometimes they are desired, but in our case, it led to decrease of viscosity of the whole system. So the whole system was not stable in the end. So we have to think about it, how to use these phenomena in the best way to improve stability, adjust viscosity, to adjust controlled release, and generally the main function of polysaccharides is to thicken the continuous phase, and while the protein actually stabilizes the droplet interfaces. Also, one very important aspect of this topic is our films and coatings based on this protein. They are flexible, biodegradable when cast or dried from solution, and those films can be used as oral dissolving films, coating layers for particles, also tablets, and or mucoadhesive delivery systems. These films offer good barrier properties, bioadhesion, also mucoadhesion, and the potential for sustained drug release. It's very important to say that compared with other biopolymers such as gelatin or chitosan, these films provide a plant-derived alternative with tunable mechanical and hydration properties, which can also expand its role of this important raw material in drug delivery. Eventually, I will say just the main advantages but also some limitations of this protein. Overall, this protein isolate is natural, biodegradable, biocompatible, multifunctional, as we saw, and relatively inexpensive. Its properties can be tuned through environmental conditions or interactions with other polymers, especially with polysaccharides. But, however, of course, there are always some limitations. This protein isolate is sensitive to pH and ionic conditions, which can lead to aggregation, undesired aggregation. Sometimes it's something we want in gelation process, but sometimes it's not desired. It can lead to instability. Also, mechanical strength may be lower than that of some synthetic polymers, and as it's from natural, it has natural origin. This variability between batches may be larger than when we have synthetic polymers, and sometimes soy proteins can have allergenic potential. It's rare, but we have to be aware that it's also possible. Despite these limitations, definitely this soy protein isolate remains a highly promising natural polymer for sustainable drug delivery system design. I will just say a few words about natural gums as in drug delivery. They are, as we can see, hydrophilic polysaccharides that derive from plants, algae, seeds, etc. Structurally, they are high molecular weight carbohydrates with branched, more or less, polymer chains, capable of extensive hydration and swelling in water media. And because of this strong water binding capacity, they can form viscous solutions and even gels, even at pretty low concentrations. And in drug delivery systems, they can serve as gelling agents, viscosity enhancers, stabilizers of disparate systems, which we focused on, and also mucoadhesive polymers. There are also mucoadhesive polymers that prolong residence time at biological surfaces. Some main structural features of natural gums is their main structure is polysaccharide backbone, composed of repeating sugar residues, and often with some substitution with side chains. And this degree of branching varies among different gums, which strongly influences physicochemical behavior. For example, some polysaccharides like cellulose derivatives, they are relatively linear, while others like xanthan gum have moderate branching. On the other hand, guar gum, that I will talk a bit later more, is highly branched due to numerous galactose side groups attached to mannose backbone. That branching affects hydration, viscosity, and interactions with other polymers, so we cannot think when we think about natural gums as stabilizer, we need to be careful about its branching because it acts different. They act different in terms of stabilization. Sometimes, as I said, they can even cause instability of the systems in terms of forming coacervates. Even two different natural gums can form coacervates. Not the typical example is protein plus natural gum, but we also have in the literature described interactions between two differently branched polysaccharides. In this experiment that I will tell you in a few minutes, we focused on the guar gum, which we can see its structure is a galactomannan polysaccharide composed of beta-D mannose backbone with alpha-D galactose side branches. And this highly branched architecture enables rapid hydration and formation of highly viscous dispersions in water. Unlike, as we saw on the previous slide, some other polysaccharides, it's non-ionic. So it's stable across different pH ranges, which is very, very important. Also, it's very strong thickening agent and compatible with most proteins and other polymers, so it can act as thickeners, stabilizers, etc. These are some main functions of natural gums in drug delivery systems. Through different, they have different mechanisms, as we can see, because of its swelling capacity. It can lead to hydrogel formation, drug entrapment. It can also lead to viscosity control, reducing sedimentation, also stabilizing emulsions. And it can also lead to controlled drug release, mostly through erosion controlled release. Also very important for these components, they, as we can see here, they can act, I will not say all the details, but through different phases lead to muco adhesion, as I said, is very important for increasing the residence time at biological barriers. Generally, when we talk about examples of drug delivery system based on natural gums, we can see that they can be for oral, topical preparations, also very often in amyl gels, nanoemulsions for mucoadhesive systems, and which is very important and which one of my PhD students now works on, are colon-targeted delivery because it's biodegradable only by enzymes of intestinal microbiota. So they are a good matrix for targeting colon mucosa, where it will be released, where the drug will be released from entrapped matrix microspheres and exert its effect. At the end of this background and the introduction story, I would just say that when comparing soy protein isolated natural gums, both are natural biopolymers but differ in structure and functionality. SPI is a protein with amphoteric charge and strong interfacial activity, so it's effective emulsifier and also gel forming material. On the other hand, natural gums are often anionic or neutral and primarily act as viscosity enhancer and stabilizers. Some of them can also have emulsifying capacity, but not so effective as proteins, and they provide stronger mechanical structure. So in combination, SPI, so protein, stabilizes droplet formation, while gum structure the continuous phase, and these combined systems actually are very important in developing the whole drug delivery systems. Based on this, what I said, we published several publications. As we can see here, mostly we combined. We didn't work only with protein isolate, also with hydrolyzed proteins, so with shorter with smaller size peptides and smaller size proteins, and also in combination with starches, with xanthan gum, etc. So now I will focus on our experimental work. As I said, emulsions. I have already said a lot of why they are desired drug delivery systems. They can be used as such as as drug formulations, topical, liquid or semi-solid, also as vaccine adjuvants. They can be in conventional and novel drug delivery systems, and despite their extensive applicability, they are of course thermodynamically unstable, and their long-term stability depends on the right choice of the emulsifier and the surface that is surface active and that forms films on the interface. Considering this whole green pharmacy concept, definitely there is increasing demand for naturally derived excipients, and in this particular experiments, in this study, we focused on the combination of soy protein isolate as emulsifier and guar gum as stabilizer. Also, we explored also their interactions between these two macromolecules in order to investigate their influence when they are together in different ratios, how it will affect its functionality that can be used in drug delivery systems. So I will focus now what we specifically did in this experiment. The aim of this study was to investigate the influence of the combined use of soy protein isolate and guar gum on the physicochemical properties of oil and water emulsion systems intended for pharmaceutical or cosmetic applications. We focused on evaluating how the presence and concentration, different concentrations of guar gum in combination with soy protein isolates affect the organoleptic properties, physical stability, and rheological behavior. Also, we also wanted to see the role of oil phase composition in determining emulsion performance. So we chose 3 different types of oils: linseed or flaxseed oil, jojoba oil, and silicone oil. They are chemically very different. It's interesting that jojoba oil is actually a liquid wax. It's not a classical triglycerides. It's a wax, so it has different chemical structure. And of course, silicone oil, as we all know, is synthetic, very hydrophobic oil that has completely different properties from these natural oils. So we wanted to compare the influence of these oils on stability and rheological properties, and also we assessed how variations in guar gum concentration modify the viscoelastic and thermorheological properties of the resulting systems. Thermorheological, I will explain later, but it's very, very important aspect of pharmaceutical systems, how properties change with difference when we differ the temperature. Also, we wanted to evaluate the interactions between protein and polysaccharide components, and to see especially their final effect on thermorheological phenomena. And overall, this study aimed to identify optimal conditions and concentrations of both components, and to optimize it in terms of emulsion stability, rheological performance, and in this way supporting the development of effective and sustainable emulsion-based formulations in the line with principles of green pharmacy. As we can see here, we prepared oil and water emulsions using soy protein isolate as natural emulsifier. We had 3 different oil phases, as I said, to evaluate their effects. And first, we of course hydrated protein to obtain a protein dispersion. Guar gum was separately hydrated to form a polysaccharide solution. After 24 hours of hydrations, we added as we can see here, appropriate amounts of these dispersions, solutions, oils, and we produced emulsions using rotor-stator system, ultra-thorax, and also in pilot experiments. I will not talk a lot about it. We also optimized the rotation speed, the time. It's very important. We cannot use it for too long, also not for too high rotation speeds, because it can denaturate proteins. So it's very, very difficult to optimize all these parameters. And eventually we explored the main aim was to explore the influence of guar gum concentration that was in the range from 0.01 to 0.15%. Also, the physical stability was assessed using some conventional stress tests like centrifugal stress testing, also temperature cycling between freezing and room temperature. Also, the creaming index, very important parameter for emulsions, was determined after storage to quantify phase separation during aging. Also, we performed visual inspections, of course, but the main part of our research was assessment of rheological properties. 24 hours after preparations, we performed flow curves experiments, standard conventional rotational rheological tests, where we increased and decreased shear rate to determine viscosity behavior and structural breakdown or recovery. Also, oscillatory rheological tests were performed to evaluate viscoelastic properties like amplitude sweep to identify that linear viscoelastic region, and then frequency sweep tests to determine elastic and viscose moduli and the whole viscoelastic nature. And the main, I would say, the main part was thermorheological measurements when we gradually increased temperature and monitored changes in viscoelastic moduli. This actually allowed evaluation of temperature-dependent structural transitions and thermoresponsive behavior that is very relevant not only for storage but also for application conditions. As we can see here, the main results, all prepared emulsions were homogeneous immediately after preparations, with colors ranging from milky white to pale yellow, depending mainly on the oil phase used. Also, the slight color differences were attributed to actually intrinsic colors of oils. When we look at pH values, they were in a narrow range around mild alkaline values, around 7.7, 7.9. It is slightly above the physiologically pH value of the skin, but we all know that the skin possesses buffering capacity and tolerates the formulations in this range, suggesting its suitability for topical use. Regarding physical stability, emulsion stabilized only with soy protein isolate showed clear phase separation after we can see here phase separation by the addition of guar gum led to stabilization of the system, especially with silicone oil that we can see that showed after 7 days no signs of phase separation at all. So the creaming index was 0 after 7 days. This is very important, these rheological parts, because as we can see from these flow curves, all emulsions exhibited non-Newtonian pseudoplastic flow behavior, meaning viscosity decreased with increasing shear rate. And this is typical for polymer-containing emulsions and reflects progressive alignment and deformation of this droplet polymer structure under shear stress. That is desired property of topical formulation, because it means that the application of some force it's easily spreadable, and so it's desired for topical application. Also, a particularly interesting observations was this rheopexy. This was unusual, but it's evidenced by hysteresis loops where the downward shear curve lay to the left of the upward curve. Usually it's different, it's thixotropic, but here we obtained rheopectic time-dependent rheological behavior, which actually indicates that this shear promotes structural buildup rather than breakdown, likely due progressive association or alignment of guar gum and protein networks during the formation. That's very interesting. It was described in some previous previous publication, but it's something that can be used in terms of development of drug delivery systems. Also very important, which I want to emphasize here, when we added zinc oxide to these emulsions, that rheopexy was not present anymore. And why we added zinc oxide? We added it as a pigment that is very common, not zinc oxide itself, but generally pigments are very common ingredients in cosmetic and pharmaceutical formulations. So their impact on rheology is something that we also need to investigate. Here we can see, after fitting these flow curves, we actually confirmed what we saw on these figures. We can see that consistency index was higher when we decreased the shear rate. As I said, that's unusual, and that's another evidence of rheological behavior. Also, we can see that with addition of guar gum, usually this N index, it's actually parameter of non-Newtonian behavior. We can see that it just slightly decreased, so guar gum concentration was not the typical parameter leading to this pseudoplastic and non-Newtonian behavior. We also tested viscoelasticity of these systems. First, we can see here amplitude sweep tests results. We can see that only emulsions that contained 0.15 guar gum had this linear viscoelastic region. With smaller concentrations of guar gum, we didn't have that phenomena. And it's actually good phenomena because these systems, we know that they act some in between liquid and solid systems. Another interesting part, that addition of zinc oxide completely changed. We can see the values of these elastic and viscous moduli G prim, G second, that are much higher after addition of zinc oxide. That's also very important result. When we established this linear viscoelastic region, we also performed the frequency sweep tests, and we can see that actually we confirmed that in systems only with guar gum at sufficient concentration, this viscous modulus dominates over elastic, so they are more liquid. But when adding zinc oxide as pigment, it becomes completely different. And I would say the most interesting part and the most significant result of our study was this thermoresponsive. Actually, we confirmed that these emulsions have thermoresponsive behavior, and which is more important, it's unusual behavior because viscosity of the systems increase with the temperature. Usually in the nature it's different, but here we confirmed. On the left side we have a figure showing emulsions only with guar gum, 0.15%, because only these emulsions had viscoelastic properties, and this oscillatory mode can be done only with this kind of emulsions. And we can see that there is the crossover point indicating a temperature-induced structuring process. Actually, the emulsions transformed at some temperature point from predominantly viscous to more elastic systems as temperature approached skin temperatures. Such behavior is highly desirable in topical products because it enables easy spreading at room temperature, but increased structure, increased viscosity and retention on the skin. Also, even more important is for mucosal applications, because it also has mucoadhesive properties, and it can achieve controlled drug release. Among the tested oils, jojoba oil emulsions actually showed the strongest thermoresponsive increase in elasticity, suggesting more favorable interactions between the oil phase and the protein polysaccharide network. The addition of zinc oxide produced a major structural reinforcement, in fact, and it increased to a large amount both moduli, elastic and viscous. But what we can see on this figure, that that point where it becomes more elastic is not so clear. It also increased with temperature, but this increase was not so steep like in the case of emulsions without zinc oxide. So we can see those delta G prim. So how elastic elastic modulus changed with temperature. From considering all formulations, definitely with only jojoba oil had the best thermoresponsiveness. At the end, I would like to conclude several important things. I would say that our study demonstrated that soy protein isolate and guar gum represent an effective natural emulsifier stabilizer combination for development of oil and water emulsions with desirable physicochemical and rheological properties. Soy protein isolate alone was insufficient to ensure long-term stability, which is very important, but the addition of guar gum significantly improved resistance to phase separation and creaming through structuring of the continuous phase and restriction of droplet mobility. The threshold guar gum concentration of approximately 0.15% was identified as necessary for the formation of viscoelastic emulsion network. At these concentrations, emulsions exhibited structured flow behavior and rheopectic characteristics, indicating progressive structural organization under shear stress, which is advantageous for topical retention and film formation necessary for long-term effectiveness. The most important finding was the emergence of thermoresponsive behavior in emulsions containing sufficient guar gum concentrations, because near skin temperature, those system transitions from predominantly viscous to more elastic structures, suggesting improved spreading during applications, followed by enhanced mechanical stability on the skin. Eventually, the incorporation of zinc oxide, although primarily intended as a cosmetic pigment, it markedly reinforced the internal structures of emulsion, and actually it acted as a particulate network forming component, because it increased viscoelastic moduli and reduced temperature-dependent transitions. Overall, I can say that the results of this study confirmed that protein polysaccharide interactions can be strategically used to design stable structure and thermoresponsive emulsions based on natural excipients, and such systems are promising for development of sustainable pharmaceutical and cosmetic formulations in accordance with the principle of of green pharmacy. Eventually, I would just say that these findings support the use of this protein isolate, which I talked a lot at the beginning of this presentation, but also of guar gum as multifunctional natural excipients for designing stable and thermoresponsive topical emulsions, but not only topical, it can be also used for peroral administrations with additional structuring potential provided by particulate cosmetic actives such as zinc oxide or some other insoluble pigments that are widely used in pharmaceutical and cosmetic formulations. That was what I wanted to say. I want to thank you for your attention. I would also like to thank to all funders, and thank you once again for your attention, and also to the organizer for your invitation to be the part of the symposium.
Thank you, Dr. Nevo.
Thank you.
Thank you again for sparing your valuable time, about more than 1 hour with us. And on behalf of some students, I have one query, one question.
Yes.
Actually, you see, a lot of work you have done in natural gums and natural oils.
Mm-hmm.
I mean, the problem with this natural oils and gums that their flow properties generally change on storage. Yes.
Yes.
Yes, sir.
That
Is there any small technique that every student or anyone we can follow in a lab?
Yes, that's some of the limitations of generally materials of natural origin, because that also variability between different batches, also storage temperature, etc. It always needs to have some kind of pilot experiments to see how it acts. Sometimes hydration process is much better in cold water. Sometimes it needs heating. So I always suggest also to my students, when they want to focus on one, for example, one natural gum or one protein isolate, not to test all of them because they are very different in their behavior, to one and to really know it very well. If it needs to be dispersed in cold water or in hot, also to take care about the storage conditions. Very important is also to the storage away from humidity, also drying before experiments, and always, of course, that's a standard part of experiments, always to first investigate the humidity percentage in the raw material, and definitely to standardize all the conditions they can, and then the results will be reproducible when we can control humidity, temperature, and also to know very well this system to choose the best conditions for the whole for all our components of, for example, this emulsion system.
Oh, yeah, great. So, Miss, you are asking to run a few pilot batches, right?
Yes, definitely. And to get to know your starting material very well, to know everything about it, and to focus on one or two, not too many of them.
Okay, dear participants, is there any question? You please text in the message box, or you can personally message me. Okay, Nevo, there is one more small question. As I saw, there are several gums here working. Can you suggest what kind of the gums can be used for the topical use, and what kind of gums can be used for the internal use? Suppose
Yes,
creams or yeah.
That's interesting question. It also depends. if we want emulsion systems, then we should use the natural gums with established also surface activity. For example, guar gum is pretty good choice. Also, tragicant gum arabic, but for example, xanthan gum is not surface active at all. It does not absorb on oil-water interface. So it's just useful for viscosity as a thickener, actually, but it does not influence so much internal structure at the interface of emulsion systems. So they're safe, and they definitely can be used for internal use, for peroral use, and also topical use. There is no limitations considering that aspect of safety. But something that I would suggest to always check is how branched. Is it highly branched or slightly branched? That because it influences the complete stability of the system. Usually, those highly branched forms more viscous matrices, and that matrix that is more viscous, it's, I would say, more suitable for peroral use because it contains more stable, for example, microspheres. As I said, for example, with guar gum for colon drug delivery, and because they can really resist the harsh environment of our gastrointestinal tract. So I would always choose something more viscous and at higher concentrations for peroral use, while for topical use, it should be used at lower concentrations only to obtain enough sufficient stability without changing without sustaining drug release so much. So we can definitely use them at lower concentrations.
Yeah, thank you, Nebo. Again, your highly and thoughtful insights. Definitely, we'll work on it and we'll convey the message to our students, those who are working on this area. And I will ask your support also to help our students in this regard. And once again, thank you. I think this is the second time. Last year also, you are also one of the speaker for us. And
Yes. I'm really grateful for your invitations. I really hope, as you said at the beginning, maybe some next time in India in person. So why not? It would be my great pleasure. But of course, it's always my pleasure to contribute. I wish you also the very successful symposium. Also, good luck to all the students, and I hope they will hear something new, learn something new, and so good luck to everyone. If there is anything I can contribute, and so feel free to also to all the participants also to contact me. I will always help if I can help.
Yeah, obviously, and actually, we got approval from ME and MHA, Government of India. So they also given the clearance for visiting in India for it as per your visa detail and all provided. And in near future, you will see how you can come to our college physically,
Yes.
so that we can have some good discussion.
It would be great. We will definitely discuss that opportunity in future. Why not? Yes. I would be grateful. Yes.
So, and again, as a token of gratitude, I will be sharing your certificates via official email ID. Within 1 or 2 hours, I will sharing to you.
Thank you very much once again, and good luck in the continuation of your international symposium. Good luck and all the best. Thank you.
Thank you, Nevo. Thank you, Nevo.
అదేంటి సార్ ఆన్లైన్ ఉందా? ఎస్ అదేంటి సార్ రాకుండా
నువ్వు కెమెరా ఆఫ్ చేసావా?
ఆన్ చేసాడు.
ఆఫ్ చే
మరి ఆఫ్ చేయరా? కాన్సెప్ట్ అయిపోయింది కదా.
ఎందుకని
పోస్ట్ కెమెరా మార్చింది సార్. సెకండ్ తీసుకుంటున్నా
పని చేయట్లేదు.
మార్చి పెట్టింది సార్. ఇప్పుడే పెట్టింది.
దీంట్లో వస్తుంది కదా.
లేదు
స్పీకర్ అవుట్ మైక్
మైక్ మైక్
ఓకే. ఇప్పుడు ఒకసారి మైక్ లో మాట్లాడండి. వాల్యూమ్ మీద ఒకసారి నువ్వు జూమ్ అయితే జూమ్ లో ఒకసారి లాగిన్ అయ్యి ఫోన్ జూమ్ లో ఒకసారి హలో నేను బయట జూమ్ లో వాయిస్
మాట్లాడండి సార్.
ఎలా వస్తుంది? వీడియోకి వెళ్ళి ఇది ఆడియో
ఆడియో ఓకే.
మైక్ ఇన్ పెట్టి
మైక్ ఇదేగా హెచ్డి క్యామ్దా?
మైక్ రావట్లేదా?
ఇప్పుడు వచ్చిందిగా. ఇది రీసౌండ్ వస్తుంది.
ల్యాప్టాప్ తీసుకోండి.
అయితే ఇది
HD కెమెరా పెట్టు.
కెమెరా పెడదాం.
హలో మైక్ టెస్టింగ్. వాయిస్ డిస్టర్బెన్స్ రావట్లేదు. అయితే మీరు ఒక్కటి ఈ కేబుల్ ఒకటి కొనేస్తే లంది కేబుల్ సూపర్ ఉంటది. లైన్ అవుట్ అండి. అక్కడ పెట్టి మెగాఫోన్ వీడియో ఓకే. కేబుల్ సూపర్. తిరగలేదు. 2 కేబుల్ పెట్టుకుంటే నీకు లైవ్ వస్తుంది. సార్ నరేష్ సార్ మీరు ఆన్లైన్ లో ఉన్నారా?
సార్ సూపర్ ఇంపార్టెంట్ బాగుంది.
ఒకసారి బయటికి వెళ్ళి సార్ ఒకసారి నేను స్టేజ్ మీద ఉంటాను. ఫోకస్ చేయి. నరేష్ సార్ ఫోన్ చేస్తాను. నన్ను ఫోకస్ చేయండి. నా వాయిస్ చూడండి. సార్ ఫోన్ కలవట్లా. స్విచ్ ఆఫ్ అనుకుంటా. నువ్వే చూడు ఒకసారి. వీడియో ఆడియో 2 ఎలా ఉంది చూడండి. వాయిస్ క్లారిటీగానే ఉందా? అంటే నీకు ఇక్కడ 2 సౌండ్లు వినిపిస్తుంది, బయటకు వెళ్తే కానీ రీసౌండ్ లేదు, నీట్గా ఉంది. ఓకేనా? ఫుల్ వాయిస్ కూడా క్లారిటీ ఉందా? ఓకే, సిస్టర్కి చెప్పండి. హలో, బాగానే వస్తుంది. సాంగ్స్ ప్లే చేయాలంటే మళ్ళీ దానికి
కురసాడివిలో నా పిల్లగా కురిసెనే గాంధారీవానా కురసాడివిలో నా పిల్లగా కురిసెనే గాంధారీవానా ఎక్కరాని కొండాలెక్కి మామ.
చూసావా? అంత దాంట్లోంచి తీసుకుంటాను.
చలిని పైన ఈ ప్రేమ మంటూ నువ్వు బండి పోనే పోదమ్మా. ఈ రోజు నా కన్నుల్లో కలై బాలిందో నీ బొమ్మ, ఇదంలా నిన్ను చూడండి ఊరుగోరమ్మా. మనసిది ఏమనది చలిని పైన ఈ ప్రేమ నువ్వు బండి పోనే పోదమ్మా. ఈ రోజు రాజల్లో కలై బాలిందని బొమ్మ, నిజంలో నిన్ను చూడండి ఊరుగోరమ్మా. లవ్లీ ప్రేమ 24/7 కురిపిస్తున్నా, ఎంత నువ్వున్నా ఎవ్రీ సెకండ్ నీకై పడి చేస్తున్నా. 7